Okay, let's get started. Today we'll be talking about noncyclic phosphorylation cycle. When a plant is exposed to red light as well as far red light, the photosynthetic rate of the plant increases significantly. This happens because of noncyclic phosphorylation cycle. So here I'm drawing the antenna complex. So each of these represents a pigment molecule. Okay, when a red photon strikes the pigment in the antenna complex, resonance energy transfer occurs. Yes, Joseph?
What is resonance energy transfer?
So, as the red photon strikes the pigment, the light energy is transferred to an electron acceptor, which excites the electron. When an electron is excited, it's raised to a higher energy state, which equals instability. As the electron falls back to ground state, the energy from the electron is released and transferred to a nearby pigment. During resonance energy transfer, the energy from the electron is continually transferred from pigment to pigment. Yes, Rena?
Which pigments are involved?
The pigments that are involved depend on the specific wavelength of the photon. Since we're only focusing on red and far red photons, we're only going to be talking about the chlorophyll pigments. Yes, Daniel?
When does the resonance energy transfer stop, or is it continuous?
The resonance energy is being directed towards something called the reaction center, which is a pigment that will stop towards the reaction center, which is a pigment that will stop the resonance energy transfer. When the energy from the electron is transferred to the reaction center, the energy excites the electron. The excited electron is accepted by an electron acceptor. During the non-phosphorylation cycle, when the red photon is involved, photosystem 2 is involved.
Oh.
In photosystem 2, the electron acceptor involved is called pheophytin. The electron from here at the reaction center is accepted by pheophytin. Before we move forward, we need to take a step back. The electrons that are excited and accepted by pheophytin, where do these electrons come from? At photosystem 2's reaction center, one of the most important processes occur: oxidation reduction reactions.
Wait, oxidation is lost, reduction is gained.
Yes, when a molecule is oxidized, it has lost electrons, while a reduced molecule has gained electrons. So at the reaction center, photosystem 2 is able to oxidize water into protons and oxygen. As water is oxidized, it loses two electrons, which is used in photosystem 2. But this is one of the most important processes because photosystem 2 is the only protein complex that can catalyze the splitting of water molecules. Yes, Joseph?
Why is that so significant?
Water is highly electronegative, meaning that it has an extremely high affinity or attraction for electrons. The oxygen in water wants to maintain its affinity for the electrons and sharing in a sigma bond with hydrogen. But photosystem 2 is able to become more electronegative than oxygen, which attracts and removes electrons from the water to produce protons and oxygen. Now, when the two electrons from water splitting is accepted by pheophytin, is pheophytin now oxidized or reduced?
Reduced.
Yes, because pheophytin has now gained electrons. These electrons then can go through something called the electron transport chain, many times just called ETC for short. During the ETC, the electrons are passed from molecule to molecule through a series of oxidation reduction reactions. The ETC in photosystem 2 consists of plastoquinone and the chitochrome complex. During the ETC and chitochromes are easily reduced because chitochrome complex is highly electronegative. Thus have a high affinity for the electrons. Now the last step in photosystem 2 is phosphorylation. Phosphorylation meant the addition of phosphate groups to ADP to produce ATP. So where and how does this happen? Okay, so when the electrons are going down the electron transport thing, the potential energy that is released is used to transfer protons from the stroma to the lumen side of the thylakoid membrane. So what does this result in? Yes, Nathan?
Large concentration of protons in the thylakoid membrane?
Yes, because protons are actively transported from the stroma to the lumen, the pH in the stroma increases to around 8, while the pH in the lumen decreases to 5. Now the protons will want to leave the lumen and into the stroma because of the proton electrochemical gradient. But because the thylakoid membrane is a lipid bilayer, ions like protons are basically impermeable. The protons are able to follow this gradient through an ATP synthase, which undergoes a conformational change in phosphorylase ADP to synthesize ATP. Now, we're not quite done yet. Non-cyclic phosphorylation cycle is a unique cycle because it is the interaction of two different photosystems as a result of two different photons striking the plant zone. We will further talk about photosystem 2's involvement in class, photosystem 1's involvement in class tomorrow. All this information about the photosystem function as well as our organization was all very recent. In 2007, Dr. Guardian was able to confirm the organization of photosystem 1 and photosystem 2 in depth in red algae plants called Cyanidium caldarium. Many experiments and information regarding the photosystems were mainly with cyanobacteria. But this paper was interesting because Dr. Guardian and his team were able to witness the function in much higher order plants.